this is this is Thursday's second talk. We have two more talks on Friday. Friday lunchtime, there's Marco Mercati, who's a new lecturer at Imperial. And then in the afternoon, we have Dr. Giulio at 5:30. Today we have Professor Vlad Vyazovskiy, and I'm really happy that he was able to come from all the way from Oxford. He did some research under Alexander
Borbély.
Sorry. And afterwards he also did some research under Tononi and Cirelli. He's currently a lecturer at Oxford. Yeah. So
not really, but it does.
Oh, and associate professor at Oxford. Is that correct?
Okay, fine.
Right.
Thanks.
Okay. Without further ado, let's begin.
Mm-hmm.
Thank you.
Okay. Is the microphone working? Yeah. And do I have a laser pointer, Giulio, by any chance? It's fine. I can show like this. Thank you very much, everyone, for coming. It is a very interesting building, very interesting setting, and probably these key words like sleep are not often said in this setting. All right, so I would like to give a brief overview. So this is what I will be talking about. I will, for some of you, I will remind. For some of you may learn that. Sleep is homeostatically regulated. I will talk about that process, and I will mostly talk about a local aspect of sleep regulation and the relevance of that for understanding of sleep function, and also for conditions when sleep is disturbed. Let me start, so I'm not going to go too far much in philosophy, but I would like to cite this very nice review from Nathaniel Kleitman, who is one of the founders of Sleep Field. And here I just want to remind that although we are interested in sleep, it is very important to give equal attention to wakefulness. So the way he put this is the following. We are familiar with the events transpiring during our wake state as against the oblivion of sleep, and familiarity breeds contempt. Thus one hears of the mystery of sleep, but not of the infinitely more complex nature of the analytical integrative performance of the nervous system during wakefulness. And this will be a recurrent theme in my talk, that we really cannot understand sleep without looking at wakefulness and what happens during wakefulness. The starting point of my talk will be about this dilemma, where sleep starts and where it is regulated. And really the sleep field started from pioneering findings by Baron von Economou, who happened to live during this period when the famous encephalitic lethargic outbreak occurred in Europe that affected hundreds of thousands of patients. Many of them died, but many of them survived, and interestingly, many of them developed Parkinson's disease later on. So this is quite a remarkable paper from 1930s that I really enjoyed reading, and this is when he suggests this idea that there may be a place in the brain which is key for regulating sleep and wakefulness. So I like that he kind of compared this with the possibility that there is a center of life itself in the brain, which we may discover by searching for where sleep starts. So there is a lot of very interesting literature which emerged from this line of research. This is a very recent review who goes in some depth into what happened to patients who experienced this disease, who survived encephalitis lethargica. So the interesting issue here is that all these findings that stimulated the idea that there is a sleep center in the brain, all this occurred when there was no EEG, and in fact at that point it was not really clear whether observations that von Economou made indeed corresponded to bona fide sleep and wakefulness. Indeed, many patients who were affected by encephalitis lethargica would enter a very peculiar state of lethargy, or as sometimes it was called torpor, which may be quite different from sleep state. So it is, of course, quite a complicated phenomenon. Nevertheless, this is our current framework of where sleep is controlled and how it is regulated. So these are very famous areas which regulate sleep and wakefulness. And this is a convenient and interesting framework that many laboratories worldwide, including here in London, work on. But I would like to discuss this idea by challenging this notion that we switch between wake and sleep, which we sometimes take for granted. Obviously, animals, when they're awake and asleep, they look different. So this is the best depiction of sleeping fly I could find, which, although I'm not sure that they sleep really this way, but in this case I would be convinced that the fly is asleep. So is it really the case that we switch between... Huh? Well, we need to test that, measuring arousal threshold. Yeah, all right, and indeed, how do we define sleep? People have thought about this, and but it is it is not an easy task, because all the defining criteria for sleep we are familiar with. They are neither necessary nor sufficient. And if you start questioning, like, why is it relevant to be quiescent, for example, during sleep? Is it that you are quiet because you are sleeping, or another way around? I think this question is still unresolved. The methodological difficulty of defining sleep, again, is not a novel issue. It has been brought up many times in various ways, and it is really important for us to be open to new ways of looking at this, because indeed it is not a trivial problem, trivial issue, especially that animals sleep in many different ways. And maybe at the end we would have to define sleep by what it does instead of how it looks. And this is because sleep is a very complex phenotype. This is just to give you a flavor of the complexity of sleep at many different scales, from its very pronounced circadian regulation, such as sleep occurs for most animals at a specific time of day, which is probably significant for its functions. Sleep is homeostatically regulated. Indeed, this one may distinguish sleep from other abnormal states like coma. There are two distinct sleep states, non-REM and REM sleep, although I will also show some data that suggests that this may not be the case. Finally, there is an infinite complexity at the level of neuronal activity that characterizes sleep in ways we don't understand yet. So where do we start from if we approach sleep regulation not from subcortical regulation but from other levels? Is, for example, we can use this influential model of influential two-process model that was proposed by Alexander Borbély and Sergei Dan, who postulated that sleep is regulated by two interacting processes, process S and process C. So most of you are probably familiar with that, but just to remind you that although neuroanatomical and molecular underpinnings of the circadian process is now understood in some detail, where is sleep homeostasis still completely unknown, and it is not even known if it is in the brain. But we have a measure for sleep homeostasis, and it is derived from EEG. If you look at the EEG trace during wake and during sleep across many animals that have been studied, I'm talking about mammals, and the same applies to birds to some extent, sleep is distinguished from wake by the occurrence of slow waves. And slow wave activity is a good measure of sleep pressure, of homeostatic sleep regulation. This is our typical night. It is a profile of slow wave activity across the night. When it starts at the beginning of the night, slow wave activity is high, and then as time progresses, slow activity gradually declines. Also, people have done interesting experiments to address these dynamics of sleep regulation. For example, if subjects are sleep deprived, then slower activity during subsequent night is enhanced as compared to baseline levels. And if subjects take a nap, then it makes subjects take longer to fall asleep next night, and slow activity is decreased, suggesting that slow activity, at the very least, reflects preceding sleep-wake history. So what is behind this mysterious process S process S that builds up presumably during wakefulness? So one way to address this question is to ask whether the content or quality of wakefulness has an influence on the dynamics of this process. A very simplified way of looking at this is the following. So you or any animal can spend time in a awake state, which would satisfy probably all criteria of wakefulness, although interestingly we don't really have formal definition of wakefulness. So you can do something very simple that you don't spend much cognitive or other resources, or you can you can be involved in something very demanding metabolically or with respect to synaptic plasticity. So do these two types of wake behavior matter? You can address this in various ways. So I really like to show this example of how you can change completely sleep-wake architecture by making very simple change in the animal's environment. I show you 2 consecutive days, and this is slow wave activity color-coded according to vigilant state. It starts with the dark onset, and the raster plot on the top shows when the animal runs on a wheel. If you simply give a running wheel to the mouse, it will spend awake about 8 hours. Some animals would spend the entire 12 hours before they take a nap. By simply removing the wheel, you can change this dynamics completely, and the animal goes to sleep much sooner. So obviously, what happens during wakefulness affects sleep quite dramatically. And this difference, this is very same mouse on 2 consecutive days. Such difference would be found quite spectacular if we are talking about some knockout or transgenic mice. But this may, so changes that we observe in sleep often may be related to changes in wakefulness. We also started, we developed a different task where we also give an opportunity to a mouse to perform a very simple task. So it is a view from above, where the mouse goes back and forth. It has to do nothing but touch the screen, and then it goes back and intermittently gets a reward. So it is a motivated behavior, clearly, but this is also something that can completely override both circadian drive for sleep and homeostatic drive for sleep, because this animal can perform this task also for 9 hours in a row during the light phase without taking any breaks. So sleep pressure is there is a lot of flexibility, which I find quite interesting. So for example, these are 2 animals that were given the opportunity to perform this task, and this is a light onset, and they continuously performed the task for about 7 hours, and some animals again could perform for 12 hours. So is this type of waking, which is very simple, very stereotypical, different from wakefulness if animals are doing something more demanding, and does it have influence on subsequent sleep? So we find that clearly it is the case, because if we keep animals awake for this exactly the same duration, which was matched for each individual, and then look at subsequent slow activity and compare slow activity after wake with exploration, this is our classical sleep deprivation, and wakefulness when the animals did nothing but go back and forth, then slow activity in the initial sleep was substantially lower in all individuals without exception that we studied. Slow activity sleep was less intense. So this means that the animal is that it is not simply time or duration of wakefulness that matters, but it is a content or quality of waking that matters. So, and in theory, you may imagine that you develop such a task where slow activity would be lower or would not be increased at all. So what does it mean, this increase in slow activity? So just to remind you quickly where slow wave activity is coming from, so you can, at least in ideal case scenario, track the occurrence of individual slow waves on the LFP or the EEG down to a behavior of a single neurons. So slow waves detected in the cortex with field potentials reflect the switch in the membrane potential of single neurons between depolarized active state and hyperpolarized down state. So it is remarkably little is known about how sleep deprivation affects this dynamics of individual neurons, because clearly this represents a very interesting opportunity if changes at the level of the EEG arise from changes at the single cell level where function of sleep probably are to be found. And in this case, of course, we need to find the right level of investigation, because if you go down to the behavior of individual cells, you can observe these up and down states, which I just showed you, but there you realize that it is no longer sleep. It is if sleep is defined as a behavior, then when you reach that level of individual cells, sleep is no longer there. It is a very it is an elusive phenomenon, if you wish. So how to approach this phenomenon? So here I would like to introduce this concept of local originals or original sleep, which is not a new idea. Several notable sleep researchers, as long as 100 years ago or 50 years ago, suggested that sleep is not a property of the entire brain, but there are maybe some areas which are more important in this respect than others. Only some parts of the brain are sleeping, resulting in you sleeping at the global level. There is a lot of evidence for that. I would like to mention just a few cases. For example, if you apply many electrodes on the scalp and look at the distribution of power, so these are 1 Hertz bins, during normal sleep, you can see that slow wave activity is most pronounced in the frontal areas. Also, after sleep deprivation, frontal cortex responds strongest to this challenge. So definitely you can immediately detect some regional aspect of sleep regulation, even at this relatively crude level. But recordings with small electrodes and even in human patients reveal that sleep is much more local than it was thought, such as if you implant many electrodes in patients with drug-resistant epilepsy for diagnostic purposes, and the patients spend time in the hospital waiting for seizures to occur, and this is when they are very happily participate in experiments. And this is how whole night polysomnography studies have been performed, which reveal that slow waves, they very often occur asynchronously between cortical areas, like in this case, when you can see both at the level of EEG and also multi-unit activity. So this is the left premotor cortex, this is the right premotor cortex, and when slow waves happen here, the contralateral area is virtually awake. So there are numerous examples of the same phenomenon in animals. Again, here you can see 4 consecutive slow wave events when with 3 electrodes about 1 millimeter apart. So here all 3 experience a slow wave, but just a few seconds later, interestingly, only slow waves occurs locally in just one area, and if you were not recording from this area, you would assume that there was no slow wave happening at all. So although we do know that transition from wake to sleep happens quickly, and so it may differ between animals and humans, but if you look at the brain activity at this transition, then you do see that this process may be gradual to some extent. In this case, we recorded from 4 derivations. These are local field potentials in a rat. And you can see that if you look at the front, then slow wave events start happening in the front and not on the back. And it takes maybe 10, 12, 20 seconds before slow waves start occurring asynchronously. So before transitioning to global sleep, the animal spends a short period of time in this state, sort of in between wakefulness and sleep, at least defined electrophysiologically. So also earlier studies, they suggested that sleep may occur at a single neuron level. It was demonstrated by one figure with this example when a monkey would perform a task. So this was a trial onset and individual, and this is a response of an individual neuron. And at some point, the animal would continue responding, but that neuron stopped discharging. So it was, of course, a speculation, but it was proposed that this neuron fell asleep because of tiredness, but this didn't really reflect the global behavior of the animal, who was still asleep, still awake. So what happens at the moment of awakening? So here, if you haven't seen this book by Sir Charles Sherrington, which is his probably most poetic book, you may find this very famous passage which describes the moment of awakenings in the cortex. So he wrote, "The great topmost sheet of the mass that where hardly a light had twinkled or moved becomes now a sparkling field of rhythmic flashing points with trains of traveling sparks hurrying hither and thither. The brain is waking, and with it the mind is returning," and so on. So it is called an enchanted loom metaphor. Of course, he was not correct, because we know that the brain is still active during sleep, so it is not you are going from nothing. But indeed, at the very beginning, when you return to wakefulness, you are in a somewhat different state compared to your normal awake state. So we asked this question recently, and we detected all spontaneous awakenings. So this is the LFP, this is the EMG, muscle activity. You can see that at some point the animal wakes up totally spontaneous. There was no stimuli of any kind. But if we measure neuronal activity in the cortex in these first minutes of awakening, we find that immediately after arousal, cortical activity is lower, and it in fact took several minutes before it reached average awake levels, even when we controlled for the amount of locomotion and movement. So there is again gradual process, and again it would be probably not easy to detect a precise moment of transition from wake to sleep, at least as long as we talk about spontaneous awakenings. And finally, this sort of mixed states are very typical for sleep deprivation. So there is now a lot of evidence that if you record brain activity in rested, refreshed animals just at the beginning of wakefulness or sleep deprivation at the end, it shows a lot of differences, and one of them is that neuronal populations behave a little bit like they're in a sleep-like mode when the animal is stayed awake for several hours. So you may see the signatures of increasing sleep pressure at the level of neuronal population when in an animal that is still awake, that is still awake. And finally, studies, although these studies are typically performed in head-restrained animals, where membrane potential of individual neurons is tracked, you find these remarkable examples when the animal, which at least according to the authors is awake, when it does not move, then the occurrence of these down states is very typical. It is only when the animal starts moving and arousal level increase, then cells transition into depolarized state. But it looks like immediately it goes to this default mode characterized by down states when the animal stops moving, although it stops really just a few seconds ago. So is this animal awake or asleep? So this is an interesting question. So what do we learn from studying, from looking at sleep as a local process and from studying a local sleep regulation? So I would like to give a few examples. First of all, looking at sleep as a local original process is very useful for understanding many of brain disorders and sleep disorders. For example, there is a group of sleep disturbances called parasomnias, which are often characterized by occurrence of mixed states. So this can be described like in this episode of confusional arousal in a child as behaviorally when this child is moving, but it would not be responsive, and if you ask him or her later on if they remember anything, they wouldn't remember. But this is also reflected very clearly at the level of brain activity, when some parts of the brain, such as frontal cortex, shows persistent slow wave activity, which is not markedly different from before the onset of this episode, but some other areas they distinctly show arousal. So this is clear that sleep can occur at the local level, and this may result in dissociated states. Also, we can learn something from looking at other sleep oscillations. For example, sleep spindles. Sleep spindles they are this is a type of oscillations which occur at around 10 to 15 hertz, and they manifest on the EEG like bursts of activity. And it has been shown in several studies that sleep spindle activity it is reduced in patients with schizophrenia. So the important point here is that all this data are based on EEG, on scalp EEG recordings, which of course does not allow us to look deeper at the more local level. So when we use local field potential recordings together with EEG in our mice, you can find quite remarkable cases like this one. So the first 4 traces are local field potentials recorded in the somatosensory cortex, and this is a classical EEG, frontal EEG reference to the cerebellum. In fact, these channels are also not really local potential because they're also a reference to the cerebellum. Anyway, spindle-like events may occur at one area. This is somatosensory cortex, without even a tiniest trace of spindling on the frontal EEG. So this means that we probably have no idea about the actual extent of sleep oscillations, such as sleep spindles, and this may have interesting implications for our interpretation of data. For example, we recently published this paper where we looked at GLUA1 subunit of glutamatergic of AMP receptor knockout mice. They have interesting behavioral phenotypes, but we looked at their sleep, and we found that if we compare spectra between wild types and knockouts, looking at the frontal EEG, then they had this marked suppression of spindle activity, and visually it was absolutely impossible to find any spindle events. So now we started recording local field potentials in these mice, and we were quite surprised to discover that at the local level, both wild-type mice and GRIA1 knockout mice do have local spindles. Again, although we completely replicated this result, suggesting that, again, our observations, even in patients with schizophrenia, that they lack sleep spindles may be completely untrue. But on the other hand, they have potentially some very interesting disruption, probably at the higher network level, which prevents them from manifesting global spindles. Another example that I would like to give is our habit of subdividing sleep into 2 distinct states, non-REM and REM sleep. So again, it is a very convenient convention, and indeed there are good reasons to tell that there are 2 sleep states, which is quite trivial. So these are EEG spectra during waking, non-REM and REM in a mouse. If you compare the blue line and the pink line, these are non-REM and REM, and you would say that based on the EEG signal, clearly these are 2 distinct states. And the same applies to humans. So if you compare the bottom trace and the top trace, so there are many differences between states. So for those who don't know, humans enter REM sleep about 4 or 5 times a night, and mice or rats may enter REM sleep many times. So it is a phenomenon which is not insignificant. This is an example in a rat of a 6-hour recording. Again, a color code, slow wave activity. Blue is non-REM sleep, and you see these excursions downward. This is when slow activity in the EEG disappears, and these are REM sleep episodes. So we have some ideas about generation of REM sleep. So it is believed that the subcortical networks which are involved in REM sleep control, they may to some extent overlap with those responsible for regulation of wakefulness and non-REM sleep, such as some hypothalamic areas. But SLD, which consists of glutamatergic neurons, and the pons is considered to be one of the important areas in REM sleep control. But the question is, okay, are they really as different as they seem, or do they share similarities? So you would argue that looking at brain activity in this case is not enough, because REM sleep has important peripheral components, and one of them is famous muscle atonia that is found during REM sleep. So if you record an EMG carefully, you can see it very nicely. So this is one of our recordings. When you use this kind of electrodes, and this is what happens with EMG variance between non-REM and REM sleep. So if you look at the magnitude of change, it is quite substantial. So there is, although the animal is asleep, there is a very, very significant change in this variable, the EMG variance, and this is the average profile of EMG variance at the transition from non-REM to REM. However, the question remains whether it is, first of all, whether it is specific for REM, and probably it is not really the case, because even human studies suggest that muscle atonia during non-REM sleep is not uncommon. So some peripheral manifestations of REM sleep may be present even during non-REM sleep, even during very deep stages of non-REM sleep, like was shown in this study. And if you look at the homeostatic response to sleep deprivation, then you may see that deep sleep stages, such as stage 3 and 4, so now it is combined in the same stage, or stage 2, and REM sleep, they may behave very similarly. Such a slow wave activity after sleep deprivation may increase across all sleep states, which led Alex Borbély to suggest that there may be some features of unitary state which are shared across all substates of sleep. And finally, the recent findings suggest that occurrence of slow waves during REM sleep is more a rule than exception. These are some examples from several cortical areas which show that slow waves can occur during REM sleep in some areas which would be almost indistinguishable from non-REM sleep. And this is something we can see quite regularly in all our mice when we record local field potentials from the somatosensory cortex. These are 3 channels. These are multi-unit activity from the same electrodes. And these are EEG signals. Especially in the occipital EEG, you can see continuous REM theta activity indicative of REM sleep, while some small islands in the cortex, like in the somatosensory, they happily remain in a persistent slow wave state. So it is which kind of challenges this notion that REM sleep and non-REM sleep are global states. This is how it looks on average, the spectrogram at the transition from non-REM to REM at the level of the EEG, where you can see familiar to many of us transition from slow wave dominated state to theta dominated state on the EEG. In the somatosensory cortex, REM sleep, defined electrophysiologically, basically never happens. So you can see this occurrence of spindles at the transition, and then slow waves persist. So does it mean that some areas do not need REM sleep, or they're not capable of producing REM state? And this reminds this interesting model of interesting idea of sleep being a default state. According to this view, slow wave activity can arise spontaneously from a network, like in this so-called cortical slab preparation. When it is differentiated and doesn't receive any inputs, then slow activity can persist for days and weeks, arising from just from the connectivity. So I'd like to add and just mention a few things of what is the relevance of this for our understanding of brain disorders. I would like to mention specifically depression, which is a very interesting condition from the point of view of changes in sleep. So in fact, it was suggested that if a depressed patient attends to a doctor and does not report any sleep problems, then probably the diagnosis is wrong. So depression is very typically associated with shortened sleep and a significant advance of the occurrence of first REM episode. At the same time, slow wave activity in depressed patients is typically reduced, which made some people to suggest that the sleep and depression may have some similarities to aging phenotype. So such a marked change in sleep in depressed patients stimulated this interesting provocative hypothesis, which was suggested by Alex Borbély and Anna Justice, who suggested that process S is deficient in patients with depression, and by bringing this process back upward can improve the symptoms. So indeed, as we know, even in very severe cases of depression, when no drugs help, just one night of sleep deprivation can produce a remarkable improvement in depression symptomatology, suggesting a very interesting relationship between mental disorders and sleep regulation. It is probably not only the case about non-REM sleep and process S, but it is probably also about REM sleep and also circadian process, which is, of course, a hugely important process in regulating our sleep and wakefulness. It was noted based on a number of studies that depending on how long sleep is and where exactly it occurs, it may be either antidepressive or depressogenic, such as the worst sleep is when you sleep the entire night or when you sleep just at the very end of the night. And this, if you remember, this is the circadian drive for sleep is strongest, and this is when REM sleep is predominant. But if you sleep early in the evening, or if you just skip this last part of sleep, then this may be antidepressive. So these findings suggest this so-called wake therapy is a very interesting approach to treat depression, but it also represents a very interesting opportunity to understand the place of sleep in this case, and in terms of what is it about sleep that is important for the brain and for brain health. If we talk about drugs, SSRIs and monoamine oxidase inhibitors are often chosen as a treatment for depression, although they clearly help many patients. If you look at what these drugs do to our sleep, it is quite striking and perhaps worrisome. So this is the profile of slow wave activity in a normal subject, when you can see dips of slow activity when they go into REM sleep. And this is completely abolished when patients are experiencing treatment with antidepressant, meaning that they enter a very peculiar sleep state, which does not really satisfy criteria for REM sleep, but is it a normal non-REM sleep at all? So these findings suggest that indeed we should start looking at sleep and wake and non-REM and REM sleep not as distinct states, but we should appreciate that there is a lot of overlap between states. So it is mostly relevant for understanding sleep in disease, So as was suggested by Mahowald and Schenk, that the boundaries between states are fluid, and this may be exactly the reason for occurrence of sleep disorders, but it could be a unique and important property of our normal sleep. So we, rather than looking at sleep, rather than looking at non-REM sleep and REM sleep and wake as something that we already understand, maybe we should also look a bit deeper to understand the complexity between within the states. So again, I would like to bring up this abstract from Kleitman, and where he cites Oscar Wilde that the true mystery of the world is probably the visible, not the invisible. Thank you very much for your attention.
So much for your talk. I'd like to present you with a small letter of appreciation. Yeah, is there any question?
Penultimate slide is your overlapping your Venn diagram. So they put sleepwalking, for example, at the intersection between wake and non-REM. But why couldn't you put it at the intersection of, why can't it be a REM disorder rather than a wake non-REM?
So sleepwalking, yes. As far as I know, sleepwalking always starts from non-REM sleep. But then, yeah, I totally agree with you. Yes, yeah. Between wake and REM. Well, in this case, if REM sleep, yes, you're probably right. Yes, I think we need to be. Yeah, exactly. This could be a form of REM sleep without atonia. But I think the reasoning for this is that it starts from non-REM sleep, and then you engage in a behavior which resembles wakefulness more than REM. But yes, this is, I totally agree, this is to some extent arbitrary.
So from your experience, Vlad, how many types of sleep in your research would you speculate you could classify?
Types of sleep? Mm-hmm.
Non-REM and REM, with how many, if you had to speculate, would you say that, are there like 10 different kinds of
No. Well, maybe we should look at this like a hierarchy. So there is just one state, sleep.
Okay, yeah. That's what I was going to ask instead.
Yeah.
It's more useful to just say it's one type of sleep.
Mm-hmm.
Or do you think that's too extreme?
This is a tricky question. I think it really depends. Like if you measure, if you apply, so let's test a hypothesis that there is more than one type of sleep. So I think it is a testable hypothesis. So you need to come up with some criteria, and then you need to scrutinize this and try to reject this hypothesis. So if we apply traditional defining characteristics of sleep, then both non-REM and REM sleep would probably satisfy equally well. Right, so including arousal threshold, which may be similar between phasic REM sleep and deeper stages of non-REM sleep. So this is when we need to know more than behavioral criteria or responsiveness, and this is when we need brain activity. And this is when the simplest approach, and I think we should probably stick to that, is subdividing this into non-REM and REM sleep. But from there, I don't think we need to create a huge array of many substates. It just does not help. Rather, we need to think of better ways how to measure this complexity that I don't really see yet how to do that.
Yeah, I compliment you know this perspective.
And
I think you made an excellent case here that distinction between non-REM and REM is more and more arbitrary, because if you look smaller cell, you lose it, you have local sleep, you have very fast transition sometimes, you have sleepwalking, where you are supposedly non-REM, but you go around and do stuff in a drive car. you know, you should divide between REM and non-REM, and by non-REM, most people understand slow wave sleep, right? Non-REM 4 is what most people study. And yet, 80% of our sleep is non-REM 2, which nobody pays attention to. So the point is, why do we bother? Why do we still bother with REM and non-REM?
Why do we bother?
Yeah, why do we still stick to it?
Yeah, I mean, for example, you see,
Well, this is not, you take them out, you have you have to completely forgo REM sleep for years. You can take them out for 30 years, go REM sleep for 30 years.
yes,
What
okay.
does it tell us about REM sleep?
Yeah, Giulio, you're bringing a very, very important point, especially when there was this recent study from about seals who go into the water, they don't show any REM sleep, yet they come back, they don't show any rebound. So this is an interesting observation which suggests that if you're not deprived of something, but you choose not to have it, you probably don't need to compensate it. In this case, I don't exactly know and don't really understand what happens here. So it is possible that this state is a mixed state in some ways. So maybe REM sleep is so important that it also happens in some form that no longer satisfies our criteria for REM sleep, and therefore it can happen even at this, I don't know, local level. So therefore I think we still should bother about this as a part of the reality. We just should not we should find a better approach to which takes into account complexity within states. So this is what I'm trying to.
I think it is a very interesting intellectual exercise in trying to redefine the whole sleep without EEG. Just you sit down there and say, how would we redefine it if we didn't have EEG today?
But you know that even in flies now there is a paper that shows slower activity.
Yeah, I think that's the wrong approach. There's no point.
Why? It shows slow waves in Drosophila. It shows sleep homeostasis.
There is body temperature, there is skin conductance, there is penile erection. So what is the function of penile erection? Are we studying the function of penile erection during sleep? We don't. So
I don't know. Maybe we'll discover one day.
I mean, I know what the function of penile erection is, but not during sleep.
Or dreaming. You didn't mention dreaming.
Dream, exactly. What is the function of dream?
On this note, if there's no...
Wait, yeah, there are no. Okay, let's take a few other questions. Yes.
Yes. Sorry, you touched upon this in your presentation. I'm not sure. Well, I just asked it anyway. Anyone else in the audience probably. It's just a curiosity that I have. I'm not an expert. I'm not in this field at all. So there was one slide where you were showing that it takes a few minutes to be fully awake when you wake up. It was 6 minutes, I think, on your slide. And everyone knows that you go through like 3-4 cycles of full REM, no REM sleep in the night. So what happens if you wake up during the night in terms of how much you will feel refreshed at the end? Does this 5 minutes of interruption of this cycle disrupt the whole process, so to speak? So if I sleep 7 hours and in the middle of the night I wake up and go to the toilet for 5 minutes, one night and the following night I don't do the same thing. Are there any studies that you are aware of that show that you shouldn't drink before going to sleep, in a sense?
Yeah, there is clearly an influence of preceding sleep-wake history on sleep inertia. So probably what you're describing is called sleep inertia. So it is this period of time that may in humans last like half an hour or even an hour before your performance is completely restored. So first of all, preceding sleep-wake history would affect the dynamics of sleep inertia. On the other hand, of course, so I think your question is, does this interruption of sleep affect performance or?
It's more, does it sort of ruin
Yes,
our,
yeah.
process of restoring ourselves? Or I think it does, but what is
Yeah. I don't think an answer exists to that. So there are a few studies that suggest that experimental sleep fragmentation in animal studies may disrupt certain aspects of cognitive performance. The best evidence probably comes from sleep apnea patients who may have these periods of apnea quite a few times per night, and they usually have increased daytime sleepiness. So there is a good, probably some reasons to believe that this is bad, but of course patients with sleep apnea may have other issues in addition to this interruption. If this does that answer your question?
Yeah, that's specific. We always see these perfect cycles,
Mhm.
right? And someone recorded them. They
Yeah, but spontaneous awakenings are very typical, both in rodents. So they may every minute or two they can show what is called brief awakening, and also humans may also wake up 29 times per night. I don't know about flies. Probably they also show some twitches. So I think it may happen, and we don't really know if it disrupts. It is a part of normal sleep structure to some extent.
Just to add to that, we had a lecture on sleep and sleep apnea by Professor Morel. She's a clinician. She was a clinician. The videos will be uploaded, so if you want to have a look at the graphs, they're there. They will be there.
I have a technical question. When you're measuring with a cortical EEG electrode in that study, first, what area of the cortex do you think you're recording from directly? And secondarily, what percentage of pyramidal neurons are contributing to that signal?
I think so. If I have an electrode, let's say in the frontal cortex, and as a reference electrode in the cerebellum, I'm recording from this entire area, so it could contribute to the signals that I pick up.
But a simple calculation would tell you that you have a handful of pyramidal neurons would be sufficient to give you a couple of hundred microvolts in your EEG.
Mm-hmm.
Which means when you're measuring these EEG
Yeah.
signals to reflect global state, a tiny percentage of
Mhm.
the cortex is actually contributing. Tiny.
I don't know. How would you measure? I mean, you can again do some experiments. You can make a transaction. There is also volume conduction that would, of course, contribute into the signal. So I think it is. I don't really know what I'm recording with the EEG. So this is as simple as that. You can see a combination of some local signals. You see a contribution of distant signal. It gives a good idea about the global brain state, but again, when I say global, I don't know exactly what I mean by that.
Yeah.
You were talking about depression before and how some sort of sleep therapy can be
Wake therapy. Mhm.
and what does it
The mechanisms for that are completely unknown. Indeed, this wake therapy can, as I mentioned, improve depressive symptomatology. The problem is that as soon as you take even a short nap, your symptoms come back. So I think it is unknown what are the mechanisms. It could be, there were ideas about synaptic plasticity, there were ideas about monoamines which are elevated by that. So this works in some ways similar to monoamine oxidase inhibitors or SSRIs, which increase the availability of certain arousal promoting catecholamines, which may contribute to the improvement.
Okay, but the therapy implies staying awake or finding like better sleep hygiene.
Stay awake. So it is stay awake. So what can make your symptoms better if you like skip every second night, or indeed if you time your sleep carefully, that you don't sleep during these early morning hours.